All right, so welcome everyone. My name is Margot Gia, and I work as the Natural Resources Planner for Windham Regional Commission. Tonight I'm on the steering committee for the Green River Watershed Alliance. And the Green River Watershed Alliance is a group of community town representatives from Guilford, Halifax, and Marlborough, as well as residents from those communities and representatives from regional and statewide organizations who are invested in water quality. And the group meets and really got together originally to focus on the issue of flood resilience in the Green River watershed, and has really worked on education and educational opportunities for people to learn more about water quality and flood resilience and what we can do as a communities to look at watershed issues from this larger scale and not just the microcosm of our right where we live or right where our community is, but really looking at how what happens up in Marlborough, up in the headwaters of the Green River, really does translate down all the way through to, into Guilford, into Halifax, and then into our neighbors to the south, where half of our Green River watershed lies across the Massachusetts border. So just a few housekeeping things for this evening. We're just going to ask that if you are not speaking, that you put yourself on mute so that we limit any distractions from background noises while our speaker is speaking. If you're unfamiliar with how the mute button works, if you take your cursor and just roll down to the bottom of your screen, you'll see in the far left corner what looks like a little microphone. And you can click on that and you will see like a red microphone with a slash through it, and that means you're on mute. So when you're ready to ask a question, we just ask that you unmute yourself and you can jump in. During our presentation, our speaker is going to be showing several slides. And if you have a question about something in specific that is easily answerable on a specific slide, feel free to jump in and ask our speaker at that time. If you have a question that's more involved or have a comment that's more leading to a discussion, we just ask that you wait and hold on to those questions until the end of the presentation, and then we're going to have a good chunk of time available for question, answer, and discussion. If you're feeling like you may be missing or may forget your question, the chat bar is also available, and you can put your question in there. I will be monitoring the chat bar. And if a question seems like it's quickly answerable, I can jump in and ask that for you, or it's something that we can wait and it just will be there, and we'll make sure to get that answered from our presenter towards the end during the question and answer period. So with those little bits, oh, and if you're figuring and wondering how to use that chat function, again, down in the bar, across the bottom of your screen, right in the very center, you'll see the chat. If you click on that, it opens up a sidebar on the side of your screen on the right-hand side. And towards the bottom of that is where you can type in your question. And that will show up in a running list in the chat. So with all that, I wanted to welcome everyone here. This presentation this evening, as I mentioned, is hosted by the Green River Watershed Alliance. And the Alliance is, again, representative of the towns of Marlborough, Halifax, and Guilford. And we've been making inroads into making connections with our neighbors to the south, where half of the Green River watershed lies, so that we can have a fuller discussion and picture about the entirety of our watershed. This evening's presentation is going to focus mainly on the upper portions of the Green River watershed, although Ryan is also very familiar with work that is happening in our southern half of our watershed. So Ryan O'Donnell, who is our speaker this evening, we're very lucky to have him join us. He's representing sort of wearing two hats tonight. One is with the Deerfield River Watershed Alliance, and they are the primary group that monitors the Deerfield River and its tributaries, and they are working in collaboration with the Connecticut River Conservancy, where Ryan also works, and he is the water quality monitor. So he organizes all of the water quality monitoring efforts that are happening in the Deerfield, as well as in other tributaries further north from there, including along the west, the Williams and the Saxons rivers, and some of our other tributaries throughout southeastern Vermont. And then with the Connecticut River Conservancy, he is organizing monitoring that is by state, and he's looking at sites in Massachusetts as well as New Hampshire and Vermont. So thank you, Ryan, for being here this evening. And I'll turn over the presentation towards you.
Great. I think that my presentation is actually about the whole watershed, not just
Wonderful.
the upper river.
Oh, great.
So all your questions will be answered, yeah. All right, so thanks for the great introduction, Margot. And yeah, so we started, the Deerfield River Watershed Association started up, has done water quality monitoring programs on and off for a number of years since the 90s. But this most recent iteration that's working with the La Rosa Partnership in Vermont, and more recently Massachusetts Department of Environmental Protection, started in 2017. So that's where my data starts for today. So just a refresher of what a watershed is. A watershed is the area of land that drains to a body of water. So in what is my favorite diagram of a watershed, just because there's so many things happening in it, you have this river, and the dotted yellow line is the area that if a drop of rain or you dump out a bucket of water onto that land, it will end up in that river that eventually ends up in this estuary. And then you can see there's sub-watersheds where there are tributaries. And everything that is happening on the land really is connected to these bodies of water. So what affects water quality? Well, I kind of just gave you the answer, and it's what happens on the land is a big factor in that. So here I have two figures that help illustrate how different land use affects water. So the first one to the left is a series of graphs that have different land uses and different colors showing you what happens when rain falls on that land. So when we're talking about a 100% forest, all of that water either ends up in these brown colors, which is groundwater, it goes straight into the ground, or it goes into this blue category, which is evapotranspiration, which is just a fancy word combining evaporation and then the water that is taken up and transpirated by plants. Once you get into the pasture, so you're just thinking grassland, you're getting a little bit more shallow groundwater, a little bit less evapotranspiration, and you're starting to see this red line, and this red line is surface runoff. Runoff is water that once it hits the ground kind of goes straight into the nearest body of water and often takes whatever it encounters along with it. When you get into something more suburban, you're thinking houses, lawns, more roads, you're going to start seeing a lot more runoff, less evapotranspiration, and definitely less deep groundwater infiltration. And then finally, when you get to something like a city where there's very little green space, almost all the water that falls on the ground is going to go straight into the river. You're going to have almost 100 or 80% runoff, and the rest evaporated or picked up by street trees. So what you can kind of guess from this is that the more developed an area in the watershed is, the more that water quality is affected. And on the right, we can see also how development affects the water levels themselves. So we have this black line, which is the rain falling. And then in an undeveloped watershed, this pink line would be the original water level in the stream. You have a slow rise to a peak and then a slow fall. However, as you increase the impervious surface, those are surfaces where water can't get through them into the ground, like concrete or asphalt, or even compacted gravel. You end up actually with more water reaching the river. It gets there faster, and so you get this really high peak. And we call that flashy, if you think of like the word for flash flood, as a sudden influx of water. And that has a big impact on the watershed. So first, here's a look at the full watershed. And on the bottom, we have just the generic Google Maps base, which you might be familiar with to kind of help you get your bearings about the watershed. The Green River watershed is very long and skinny, which is interesting. And then we have the water lines and then also the town lines to help you figure out where the water is. So here's a look at the sites that we've monitored in the past 4 years and their locations. There is also a bacteria monitoring location kind of upstream of Greenfield proper, but it's monitored by the Greenfield Department of Health because it's where the town swimming pool is. If you've ever been there, it's actually just a dam on the river. It's not a pool. But it's, they had some big chains up a few years ago and it's been harder to track down their data. So I'm not able to use that data for this presentation, so I apologize. And then this is the Google satellite imagery, so you can see kind of the land use. We have a lot of forests up in the headwaters, and then we're getting more fields and development until we get into the city of Greenfield, which although is a small city, definitely has those urban impacts on the water quality. So here's a look at our sites. So like I said, this program was kind of restarted in 2017 in its current iteration, working with the La Rosa partnership through the Vermont Agency of Natural Resources. And then in 2019, the Massachusetts Department of Environmental Protection also started offering funding for bacteria monitoring in Massachusetts. So that really helps. The DRWA program is really a unique one because we're working in 2 states simultaneously, which has its own challenges, but also I think is very rewarding that we're able to coordinate this monitoring across political boundaries. So all of these sites have at one time been monitored for E. coli, bacteria, nitrogen, phosphorus, and conductivity. I'll talk about all of those as I get to them. Unfortunately, due to the pandemic and funding constraints, we were only able to monitor for bacteria this year. So in addition to monitoring the Green River itself, we also monitor 3 tributaries, the Pond Brook, Hinesburg Brook, and Maple Brook. And I put them in right where they kind of intersect with the river. For the main stem, well, for all of them, I have their mile number listed. And so the way the river mile works is that 0 is the mouth of the river. So in this case, 0 is the Deerfield River. And then, it goes up from there. So the closer to 0, the closer to the Deerfield River we are. And then this also tells you the area of land that is captured upstream at each site. So the lowest site is monitoring essentially 89.2 square miles. And then the years that we monitored, I don't actually know the area of land draining to Maple Brook. It's really not unique, but unique in this watershed in that it's buried under Greenfield, and it's basically tied into the stormwater system. And so the software I use to calculate this can't understand how the stormwater system is affecting what land is getting into the water. here's a map look at those different drainage areas. We worked really hard through the support of the Green River Watershed Alliance to monitor very well spaced out in the Vermont portion, specifically to get a good look at the whole watershed. And then I mentioned that I didn't know where the Maple Brook was draining. I estimated this by looking at a stormwater map of Greenfield and a topographic map. So that's my best guess of the areas that are draining into Maple Brook, but it might not be 100% accurate. All right. So first, I wanted to take a look at stream flow. One of the challenges of my job is that it's very hard to compare across years, and that's because each year is so different. And so I wanted to show, you know, the different years and what the stream flow looked like. So we only sample up to 6 times each year, which is more than many locations in our region are being monitored, but it doesn't always provide a complete picture depending on what the flows and weather was that day. So for assessment of a river or for a true comparison between rivers, we're actually aiming to capture with our samples low flows and dry weather. We learn other things from those other samples, so we don't mind capturing moderate or high flows or wet weather events. They tell us about runoff and other sources of contamination. And the low flows and high flows are determined mathematically. So the high flows are the upper 25%. They're above this straight line of all types. So the upper 25% of all flows that have ever happened. And the low flows are the lowest 25% of flows that have ever happened, which for some reason is weirdly confusing. But I put the lines on there so you don't need to think about that. So we're already probably a bit more familiar with linear versus logarithmic or exponential graph scales since we've been looking at different graphs for the past 6 months. But this one here is on a linear scale, which means that each unit is given an equal amount of space. And so we can see really how high the high flows are, but we can't really see a lot of the details in the low flows. So here's the same data, but on that logarithmic scale. So you can see that 1 goes to 10, goes to 100, goes to 1,000. And this lets us compare, not only see the high highs, but also we can see the low lows. And in particular, we can see how dry it was this year. It really was very dry. There was also a drought in the beginning of 2018, which is kind of the medium blue color. And but then it got very wet. I don't know if you remember that year. It was very dry and then it shot up and was very wet for the rest of August. It was an interesting year to monitor for sure. Particularly in the Deerfield watershed. Got way heavier rains than some of our watersheds to the north. All right, so the first parameter I want to talk about is E. coli bacteria. So E. coli is a bacteria that's found in the guts of all warm-blooded animals, including humans. And we monitor it to determine the level of risk for recreation. There are 2 kind of types of recreation that they set levels for. So primary contact, which is swimming, splashing around, getting in the water. And it's when you're likely to get the water in your mouth or other mucous membranes. And then there's secondary contact, which is more like boating or jet skiing. I mostly only put swimming on here because there's not a lot of boating happening until you get really down. There's not a lot of boating happening, but you could boat in the lower river if you wanted to. But there are some dams in your way. All right. So E. coli is what we call an indicator organism. And so we use it as an indicator of the likelihood that the water that we're testing has come into contact with human or animal waste. E. coli itself, like I said, we all have it in our guts, is not likely to make you sick, although it can. Sometimes it does. But there are other pathogens or viruses, bacteria that might be in the water that are associated with particularly human waste that are much more difficult to test for. But E. coli is abundant. It's easy to test for. We collect in this bottle, and then we get to look at it in these trays under a black light, and it gives us a number. So sources of bacteria include agricultural and urban runoff, failing septic systems, leaking sewer pipes, pet waste, and wildlife waste, etc. And what we almost always see increased levels after rainfall. And so we recommend that you don't swim immediately after heavy rains. And for the sake of our monitoring, we consider anything over 1/10 of an inch as wet weather. All right, so I'm going to present each parameter's data to you in a few ways, because each way shows you different things, but they're also imperfect. They don't show you the full picture ever. Like I said, it's very hard to compare year to year. It's very hard to compare site to site because of different flows. Also, this is less of a, because this program has started more recently, we haven't moved our sites around a little, but we didn't monitor all sites all years. So, it would be hard if you were averaging 5 sites from one year, but only 3 sites from another year. Anyway, so the first way I'm going to show you each parameter is actually these line graphs, which is an attempt to compare both year by year, split by wet and dry weather, and then also represent the data spatially. So across the bottom, we have the river mile. So remember that 0 is the Deerfield River, and it goes from downstream to upstream, or it goes from upstream to downstream, left to right, which is probably the more intuitive way that you would think about the river. So up here, we're in Marlborough, and down here, we're in Greenfield. The other thing is, for most of the parameters, you'll see just a regular average, but these are geometric means, which is just a different way of calculating an average that we use because biological numbers have really big variations that, because living organisms like to clump together. So you might see that sometimes and have wondered why we're doing that one differently. Anyway, in dry weather, we see, in general, good weather. In dry weather, we see good numbers until we get to Greenfield. The Greenfield sites are around mile marker 2 and below. As for wet weather, we can see it's really variable, and that probably has a lot to do with, like I said, wet weather starts at 1/10 of an inch, but could include up to the really heavy storms we had in 2018 that may have been over an inch of rain. So some of that variability between years may be how big the rainstorms were that we captured. But you're still seeing way higher levels in the Greenfield area, either way. And then here, this red dotted line is the swimming standard. So we also monitored 3 tributaries, and I'm just going to show the averages by year. Pond and Heinsberg Brooks are up in the headwaters, and then Maple Brook is in the heart of Greenfield. It's basically a storm sewer at this point, which is unfortunate. And we see the huge differences between them pretty consistently across parameters. And you can see this is the difference between a headwater, forested stream. There are a few houses in each, but not, a lot of impervious surfaces. And then Maple Brook is just, well, it's off the charts consistently in dry or wet weather. And then I also wanted to take a look at each site's numbers based on the flow level. If you're a volunteer, you know that you note this on your field sheet as an observation. But since the Green River has a USGS gauge and chlorine, I'm able to look at those numbers like I presented at the beginning and compare the flows of each day to determine low, moderate, or high flows. These aren't broken down by rain. So most notably for bacteria, you can see that the moderate category has like a huge range for all of the sites. I cut the graph off at 2,500 because that's where the bacteria test maxes out. But you might notice the geo means are pretty high, and that's because of math. Okay, so these high flows, especially in the upper water, captured the really, really high flows of 2018, which saw even Pond Brook and Heinsberg Brook testing pretty close to max or over the max test. And that was just because I think it was like a 2-inch rainstorm right before we tested. There's just so much water. The ground was already pretty saturated, so all of that water was bringing, everything from the land, and that included all the deer, bear, raccoon poop. All the poop got in the river at that point. So I didn't have a chance to do it, but I would like to look at this graph with those really high numbers taken out, because I don't think that's a true impression of the headwaters of the Green River. As you can see, the averages are still pretty low, but in low and moderate flows upstream of Greenfield, we're seeing occasional high numbers, but overall pretty good numbers, which I like to see. The next parameter that I'm going to talk about is total nitrogen, and this is a graphic of the nitrogen cycle. Shows you how it gets around the environment, the ecosystem. So nitrogen is one of 2 nutrients that we test for, and that affects plant growth, the other being phosphorus. And if you're a gardener, you might be thinking you always need more nitrogen, but generally water systems are limited in their nutrient availability. And so while both can contribute to an increase in aquatic plant growth or algae growth in both saltwater and freshwater, nitrogen is generally the controlling nutrient in saltwater. So once the water leaves the Green River watershed and then goes into the Deerfield and then goes into the Connecticut, it eventually ends up in Long Island Sound. And they have a big nitrogen problem down there. There's a dead zone every summer where there's too much algae and there's no oxygen and fish die, and it's not good. So even though it's very far away, it's definitely something that I'm always thinking about reducing our impact on our downstream neighbors, and that includes Long Island Sound. So total nitrogen, the total mean that it captures all forms of nitrogen, some of which include nitrates, nitrites, ammonia. Sometimes you test for just one of those, but we just test for all at once. Sources of nitrogen include agricultural runoff, wastewater, failing or leaking septic systems. And nitrogen is very mobile. It likes to just be in the water. It doesn't fall, like sink to the bottom or anything. And so we see that the levels tend to be a little bit less affected by rain than some of our other parameters. So first, once again, a look at these spatial by year graphs. And pretty good numbers until we get to Greenfield again. I did want to mention that this purple line is a suggested level. The standard in Vermont is 5, and as you can see, we're nowhere near 5, even in Greenfield. And then Massachusetts does not have a standard, a numerical standard for rivers. But this number is based on a suggestion from the EPA for the ecoregion, basically our regional ecosystem, based on the type of forest and the geology and our climate, this is what we would want to see in an undeveloped river. So this is what I like to compare our numbers to. There was one year, 2018, where we monitored both above and below the Timber Crib Dam. And what's really interesting to me is that you can see that in wet weather, there's a very slight contribution from the dam. And that means that probably some nitrogen that is stored up behind the dam is getting out and coming downstream when flows are higher. And we'll see that on the next page. But overall, you know, pretty good until even when we get to Greenfield. Could be worse. Anyway, so look at I'll look at the tributaries, same story, different parameter. Here you can see actually that when it's raining and there's a lot more water in Maple Brook, you're actually getting diluted with the nitrogen. All right, so, and then here's a look by flow level. And you can see for the most part, once again, doing pretty well. We've had some really high levels at Bear Ass Beach, which is right upstream of Greenfield. So we know from the previous graph also that the dam might be contributing nitrogen during rain events or higher water. And so what's interesting is on this and then on a river I looked at previously, generally you see that in the high flows, like it really does get diluted. But here we can see there's actually more nitrogen below the dam and then also at the state line. And so I don't know, this might be something to look into. We don't know if there's just not that much water coming in to dilute between the dam and the state line, or if there might be even another source. So that's something that I will keep in mind moving forward with the program. And here you can actually see in Greenfield, this site, the Massachusetts 2A Greenfield site, is downstream of Maple Brook. And so we see really high nitrogen levels in dry weather coming from the brook, but we don't see those high levels in high water because it's more diluted. So total phosphorus is another nutrient affecting plant growth, but it tends to control that growth in freshwater systems. If you live in Vermont, you hear a lot about phosphorus in relation to Lake Champlain because that's really what's causing their problems. However, we are seeing an increase in cyanobacteria and other harmful algal blooms in Massachusetts, and I think also up into Vermont on the Black and other rivers where there's an impoundment, where there's a dam holding back water. And these seem to be directly related not only to increased nutrients, particularly phosphorus, but also the warmer water temperatures. We're having these long, hot, dry spells in the summer that we just didn't used to have, unfortunately. So we have a graphic of all the different places that phosphorus is in the world. Like total nitrogen, total phosphorus captures all forms of phosphorus, including organic and inorganic, dissolved, suspended, etc. Its sources are also usually agricultural runoff, wastewater, failing or leaking septic systems. And then one of the interesting things about phosphorus is that it's very sticky. If you're a volunteer, you know that you collect it in a glass bottle, and that's because it would stick to the plastic and you wouldn't get it back out when you were processing it or anyway. But also it means that it really likes to stick to sediments in particular, which is you're gonna see as a big factor in the Green River watershed, especially the headwaters. And so it gets transported with sediment and the levels are definitely affected by rain and turbidity. And turbidity is how cloudy the water is. And I'll talk a little bit about it. So first we look at the phosphorus by the year and these streams, and we see once again, pretty good levels in dry weather. And so here we have two standards, or a standard and a suggestion. So we're aiming for Class A for the Green River in Vermont. And then also Massachusetts doesn't have a numerical standard, but they do have a suggestion that rivers be between 50 and 100 micrograms per liter of phosphorus, which is, in my opinion, still a little too high. So if you live on the Green River, especially in Vermont, you know that in recent years it really runs, you know, like chocolate milk. I've heard it described when it rains. And that's due to several mass failures or landslides that are active and they get reactivated every time it rains. And that's just dumping sediment into the river. And those sediments are absolutely full of phosphorus. And so we see that reflected particularly in these 2018 numbers where we had those really, really high flows in August. And here, once again, you can see there's almost no difference in 2018 between the sites up and downstream of the dam. But here in wet weather, you can see that the dam is holding back sediment and then dumping it also. So really interesting, but also that, you know, even the headwaters can be pretty high. And then we go down as we get closer to Bear Ass Beach, as more water is coming from different parts of the watershed that are maybe a little less sediment rich. And then we increase as we get back up into Greenfield. So here is where we really see those sediments, because like I said, those wet weather numbers capture those really high flows. And so you can see in dry weather, we see the thing that we're expecting, which is Pond and Heinsberg Brook are low, Maple Brook is high. However, in wet weather, and when you include those really, really, really high numbers, the average gets brought way up for these little tributaries. But Maple Brook, not being affected by, you know, landslides, it's not really as affected by rain, which is interesting. So we can see, and you know, here's it by flow, and you can see those high flows really captured some really, really high phosphorus. And we can see in this and the line graphs that even at low flows, the headwaters of the Greenfield are really struggling to meet that Class A standard. However, I don't think that that means that it's necessarily impaired or that it shouldn't be Class A. I think that, you know, the Green River is at the southern end of Vermont, and these standards are based on data collected throughout Vermont. And we know that the Green River watershed is very rich in this glacial soil that is just full of great nutrients. But I think that it means that it's a naturally more nutrient-rich watershed, that our baseline levels are a little higher than the rivers that it's being compared to, you know, further north in Vermont. So that's something to think about. However, I do think that the mass failures are contributing a lot, and some of that also is coming off of our dirt roads, which, as we know, wash into the river if it rains too much. So that is also a concern. So yeah, you can see you know, Greenfield is once again not doing well. So I talked a little bit about turbidity. Unfortunately, we only monitored turbidity in 2018, but I was able to put together a comparison between the phosphorus on the y-axis and the turbidity on the x-axis. And so a reminder that turbidity is how cloudy the water is. And in most of the rivers that I have monitored for turbidity in southeastern Vermont and even in Massachusetts, most of the rivers really never get into high turbidity, but we do see it on the Green River. It can get pretty high. So I split it into, in light blue, the upstream sites, which basically Bear Ass Beach and upstream, and then the greenfield sites based on land use, because the greenfield sites are in a more urban land use. And what we see from these upstream sites is basically a near perfect correlation between the turbidity and the phosphorus, which tells me that that's probably, that the dirt in the water is probably the main factor contributing to those levels. Whereas there's a little bit less of a perfect fit when you're adding in those urban land use factors, which might be contributing something else, you know, wastewater, more septics, et cetera. So just something that I wanted to look at, because I really don't think, I really think that the phosphorus problem that we see sometimes in the Green River is tied to these landslides. All right, so the last parameter that we monitor for is specific conductance, which is it's a general monitoring parameter. It's kind of the catch-all, and it's really easy to monitor, which is why we do it. But basically, it measures how well the water we collect tests electricity, which is an indicator of how many ions or random stuff is dissolved in the water. So sources for increased conductivity include road salt, runoff, but also geology. So like if you have a particularly dissolvable rock in your watershed, you might have higher conductivity. It tends to be diluted with rain, but the reason that we monitor it is that it may catch areas impaired by parameters we don't otherwise monitor. So it does capture phosphorus and nitrogen ions, but it also captures, like I said, road salt is a big contributor to the conductivity differences from the background levels. But it may catch metal contamination. If you have an iron seep, you would expect to see really high conductivity. So I've been saying about the specific conductance and conductivity, and they're very, very slightly different. Specific conductance, which is what we actually record, is adjusted for temperature so that you can compare different temperature samples to each other. But just straight conductivity is related to the temperature. And then here's a little chart showing the conductivity levels of different kinds of water. So we're in freshwater streams, and we are, and I would say in my experience monitoring in southeastern Vermont and western Massachusetts, that our basic levels are usually from 50 to maybe 250. So we don't usually see into the quadruple digits. Although, spoiler alert, we do see quadruple digits in Maplebrook. So
Brian?
anyway, looking at the main stem, yeah.
A specific question from Kelly Setner. She asks, do you use a particular probe to monitor specific conductance? Is that parameter collected on site or from a sample?
So we only have one probe that we use. And so we have them collect in the square bottles, like they do also for turbidity. And those are the right size, and they have enough water and allow you to shake it that you can put the probe in the bottle. And usually I or one of our interns does that the day of sampling when they get to the lab. So looking at the main stem, in general, what we expect to see is an increase in conductivity as the water picks up more stuff. Naturally or unnaturally, it's just picking up more stuff as it goes downstream. We see that in dry weather, for the most part, this weird drop off in Greenfield in 2019 is strange, but you know, things like that happen. And then you can actually see that it's more diluted, that it's lower in wet weather. But there's no like sudden spikes. So I can probably attribute this higher spike to Maple Brook coming in here and then getting diluted by the time it gets down there. Looking at the tributaries, like I said, we see up towards 1,000 in Maple Brook, which is hugely different from the like 50 that we see from Pond Brook and 100 that we see from Heinsberg Brook. And once again, what's interesting is that Pond comes in a little bit higher when it rains, but you know, maybe we're catching some of that dirt, the sediment coming down river when it's raining hard. So once again, this is really interesting. I just also did this for the Saxons River, and in every single site, it was the highest during low flows and the lowest during high flows. And we don't see that in the Green River. So I found that really interesting. And, you know, here, you know, high flows are lower than moderate flows, so we're seeing some dilution. But I think, you know, the story of the Green River is that it's just carrying a lot more stuff in it when it's raining. So that could include road salt, phosphorus from those mass failures, et cetera. So that's really, really interesting to me. So some conclusions from this analysis. In general, the water quality upstream of Greenfield is good to excellent, which I think, you know, we knew living on or near the river. The E. coli levels tend to be generally low during low flows. However, this year and in years past, we have occasionally seen high levels of bacteria at the swimming hole at the Timber Crib Dam in Guilford. There's definitely more investigation that is needed, but when I was putting together these numbers, it didn't jump out to me as a problematic area. That might also be because I deal with some very problematic areas, which I won't bring up. Anyway, the upstream levels of phosphorus are sometimes high, and we can probably pretty confidently attribute that to sediment from dirt roads and active mass failures, dumping sediment into a river. The Timber Crib Dam may be, like all dams, may be a source for nutrients during high flows. So it's holding back water, it's holding back sediments during lower flows, and then when the water comes over at a greater speed, it's picking up some of that sediment and dumping it over the dam. And Maple Brook is a serious water quality concern for the river and for Greenfield. And I do have plans working with DRWA and CRC to get in touch with, you know, the town of Greenfield and do a more intensive study. CRC actually did a study of Maple Brook in the early 2000s, and we were actually able to correct a source of raw sewage that was getting into the brook, and we were able to test for it and resolve that. But clearly there are some other issues, and so we'd like to go and do that project again. Obviously, like many plans, we were hoping to start that this year, but maybe next year is our year. So in addition to answering any of your questions, I also want to know what your concerns are about the water quality of the river, and if you have any areas that you're concerned about that I should maybe look into more or consider when analyzing the results. But I'm going to stop my screen share so I can see all of you, and I'm happy to answer any questions.
Yeah, now we can.
If you do have any questions, feel free to unmute yourself and jump right in and ask a question. Ryan, you did get a compliment on the owl. That was inquisitive there. John, it looks like you have
I
a
think
question.
I searched questions.
I do have a question. Ryan, really excellent presentation. Thank you so much. The concerns that I have are some major sites that are along the river that are actually above the Green River drinking supplies
Yep.
that are on, I believe, conservation land. And I'm just wondering about how that contributes to the difficulties and the concern about the drinking supply in Greenfield. The other thing is, I do realize that Maple Brook is underground, and I do believe there's a number of source points that are leading into that, which cause it to be poor quality as it exits into the Deerfield down below. And I really appreciate your concern about that lower stem of the Green River, because that's really the statement about what we do with the water as it passes through Greenfield, as it becomes grade A into poor quality. I'd like to ask us all to hold the Greenfield people accountable for that and figure out how we're going to address that problem. Thank you.
So Bear Ass Beach is not that far upstream of the drinking supply. Although there have been times when the levels are concerning, and luckily that volunteer is he's at the river a lot, and he's in contact with me a lot. And so it's definitely something we're keeping an eye on, because every once in a while we do see really high bacteria coming down. And I know that there are some like trash dump sites, I think that maybe that you were talking about also. And unfortunately, many of the concerns that might be from trash or metal or leaking oil, we might catch with the conductivity, but we're not testing for oil and gas. We're not testing for some of that stuff, just because it's very difficult to test for.
Yes, and
but it's definitely something. I was gonna say the one thing that might catch it is on our field sheet when volunteers are collecting samples, they are asked to make observations about how the water looks and how it smells. And so if there was an oil sheen, if it smelled like gasoline or oil, if I got a report of that more than once on a field sheet, that's something I would definitely follow up on. I've been known to drive out to sites of concern multiple times, trying to see if I can figure out where a weird smell is coming from or where bacteria might be popping up out of nowhere.
Okay, well, thank you. And one more comment is, I know there's a major site above Nelson Road in Greenfield that is in a deep ravine that when the rains come, it is very problematic. And I know the EPA is aware of it, but there's been no action on it. And thank you.
That's good to know. Not included in this presentation because I haven't done any sampling on the Green River, but I have started working with the EPA to do some stormwater testing. And so that's something that I can do and, kind of nudge the EPA if there's some sort of violation. And that tests for a whole different set of parameters. We can run out there and grab some storm samples next year. With the incoming snow, I think my sampling season, actually today was the last day we collected bacteria samples on the main stem, and I think we finished exactly in time.
Nice job. Thank you.
So we have a couple more minutes. If anyone has any further questions or observations about water quality or anything that you've been wondering, Kelly?
There we go. Did that work?
It did.
Awesome.
Yes.
I know you have your hands full with all of this chemical data, the training of the volunteers, coordinating so many logistics, and not just for your water, for one watershed either. So major kudos to you. I appreciate it. I appreciate all the help you've given me up here in the black. You have not been able to incorporate any bioassessments in this, correct? macroinvertebrates?
Yeah, we don't currently do any bioassessments. However, there was one of the prior DRWA monitoring initiatives headed up by Mike Cole, who was a former board member and who's a macroinvertebrate guy. I forget the word. Taxonomist. He did a number of studies throughout the Deerfield watershed, including on the Green River and some of the tributaries. And I think they are all pre-Irene, I believe, but the Green River did, and the Deerfield River watershed as a whole in Massachusetts, mostly focused, did very, very well. And Mike was actually really surprised at some of the rare species that hadn't been identified in Massachusetts before. The Deerfield really has some really good water in it.
Nice, good to know. Thank you.
Ryan, we have a question from Linda Lemke in the chat, and she asks, can you talk about the increased monitoring you might do next year at the Crib Dam?
Yeah, so there have been some concerns about the high levels we saw this summer, which could indicate either a new source of contamination or an old source that's showing up because of the drought. So it's hard to tell. So in addition to our normal sampling, which we can continue if we have additional support, we could do more frequent monitoring for bacteria, which would give us, like I said, a more complete picture than 6 times. If we did 12 times, that would be a more complete picture. And then also that stormwater testing, like I was said, we can go out there and do that, since I know that one of the concerns is that, are we seeing higher levels of bacteria due to an increase in village residency, right? More people are there full time than there normally are. Is it from increased septic pressure? So that is a concern. I really wish that we had the nutrient data from this year. We just don't, unfortunately. And so definitely something that will be on the list of things I check. When I get the water quality results, there's like certain sites that I immediately run to, and I'm like, what did this number say this week? So yeah, there's a number of options, and and when I get into planning, probably in February and March, I sit down and I talk with Marie about what she wants to hear, and I will sit down with the Green River Watershed Alliance and make sure that I include their concerns to the best of my ability in the program.
I have a question. You were saying that salt coming off of roads and other places contributes to I forget exactly what it was measuring. Conductivity, right? And where it flows into, where the Connecticut flows into Long Island Sound, there's also, it can be this bloom that kills animals, but of course salt water itself has a lot of salt in it, and I guess it has a very different ecosystem. And so what's causing the bloom? It's obviously not salt, right? It's something else.
No, it's the nitrogen mostly. The nitrogen and the phosphorus. The salt is more likely to have an impact closer to home on freshwater systems. And we are seeing across the Northeast, and actually across the north of the U.S., where we use road salt. We've been using road salt more and more liberally. There's not a lot of regulations on how much road salt gets put down. And obviously the focus is on safety, which in many aspects often trumps water quality concerns. But we are just seeing, especially in our lakes, increased salinity levels, which is having an impact on the ecosystem. But I can't speak specifically to the Green River. We monitored chloride a little bit at the beginning of the program, and we didn't see particularly high numbers. So we switched to conductivity because it's a little easier to do.
Wonderful. Thank you, Ryan. We've come to the end of our program time. And if you do have further questions, you can either reach out to a member of the Green River Watershed Alliance. You can reach out to me. My name is Margot. I work with Windham Regional Commission, but I also am on the steering committee for the Green River Watershed Alliance, and I can help funnel your questions either to Ryan or to other members, Marie Caduto at the state, or appropriate partners for questions that you might have related to the Green River and its watershed. So I wanted to particularly thank Ryan this evening. It was a really informative and helpful presentation to be able to see the wonderful work of the volunteers that go out and collect this data every 2 weeks during the summer and be able to see it sort of over several years. And so again, thank you, Ryan. Thank you to the Deerfield River Watershed Alliance, who organizes the monitoring in the Deerfield and its tributaries, and the Connecticut River Conservancy, who oversees sort of all of the monitoring. So thank you again. And we hope, if you are interested, I have been recording this presentation. So if you'd like to review a part of it, you can either find it on the Green River Watershed Alliance webpage, on its Facebook page, or eventually on the Windham Regional Commission webpage. So thank you very much, everybody, and I hope you have a good rest of the evening.